all, welcome to EZI. So here we are going to see in the session different questions that is certainly going to come in your biochemistry exam. So let's start our session, and the first question is, what is this glycolysis? Again, listening to all these important questions and you will have a confusion where I have to start. So will it be easy for me to start my exams, or how is this question paper going to be? So don't worry about it. We EZIners are here to help you, and the first question let's discuss in a simpler manner so that you're going to certainly understand about it. So what's this glycolysis? Glycolysis is a process wherein there is a glucose is breaking down to produce energy. So that's your simple definition about glycolysis. So you have to by heart it like glycolysis, or you are going to understand it how glycolysis is a type of process wherein it is breaking down. So here is your important key point, and where it is producing energy. So this is about glycolysis definition. Then what is it producing basically? So there is a production of two molecules of pyruvate and ATP, NADH and water. So they are producing these kind of things: water, NADH, ATP, and one more thing is the pyruvate. So this is happening in glycolysis, and where it is mainly take place in, so where exactly it is happening in the cytoplasm of the cell. So, and where it is happening in the cytoplasm of the cell, and it is not requiring any amount of oxygen. So that is about glycolysis. This much you have to know a gist about glycolysis, and it is occurring both in aerobic and anaerobic organisms. So this is a small gist about glycolysis. If you write this, you are going to definitely get 2 marks in your exam. So then, glycolysis is consisting of 2 phases. So in the first phase, what is happening? In the second phase, what is happening? So in the first phase, what is happening? There's a glucose is breaking down, and there is a molecules of glyceraldehyde, 3-phosphate in a series of 5 reactions. So in the first phase, this is happening. So this much you have to remember in the first step. And coming to the second phase, there is another series of 5 reactions wherein there is conversion happening with 2 molecules of glyceraldehyde, 3-phosphate into 2 molecules of pyruvate. So that is happening in your second series. In the second phase, these are the things that is happening. So when you're writing your exam, first you are going to write the definition, you're completing with that. Next you're writing the 2 phases. So there itself you're getting your almost your 2.5 to 3 marks is definitely coming over there. Then phase 1 and phase 2 consumes, phase 2 generates 4 ATP, and the net ATP production in the entire process is 2. If you write, it's well and good. If you don't write, it's okay, leave it. So this is about glycolysis, and when it is coming to a diagrammatic part also, you can just mention this diagram in case if you're remembering, and try to write it or try to draw this diagram 5 to 10 times so that you are remembering this and you are writing in your exam. Then in the simple way, how we are going to, what are the steps that is in the glycolysis. So in the first step, you can see the glucose is getting converted into glucose 6-phosphate. Then the glucose 6-phosphate, it is getting converted into fructose 6-phosphate. Then fructose 6-phosphate, again there's a conversion, and even there are some enzyme that is getting included, and that is breaking down into fructose 1,6-biphosphate. So these and all, when you're trying to by heart it or trying to understand, definitely you have to by heart it, because these are some technical terms that you have to write in your exam. So you cannot understand it, but if you're understanding well and good, if you're trying to by heart it, try to write it so that your spellings also will be correct in your exams. Then fructose 1,6-biphosphate is again getting converted into glyceraldehyde G3P, and dihydroacetone phosphate is also getting conversion, that is DHAP. Then glyceraldehyde 3-phosphate is getting converted into 1,3 biphosphoglycerate. So there's a conversion happening. Next, the sixth one is 1,3-biphosphoglycerate into 3-phosphoglycerate, then 3-phosphoglycerate into 2-phosphoglycerate, and 2-phosphoglycerate into the phosphophenyl pyruvate, and the phosphophenyl pyruvate is conversion is happening into the last step, that is pyruvate. So in this manner, there's a glycolysis step happening. So if you want to remember this, definitely you have to tell it orally as well as write 2 to 3 or 4 times until you're getting thorough with the steps of glycolysis. Then there is a small simple technique that you can remember this. So there's a kind of mnemonics for glycolysis. So here you can see, goodness gracious, Father Franklin did go by pickling pumpkins to prepare pies. So where? So what is this mnemonic meaning is, so goodness gracious Father Franklin did go by pickling pumpkin spice. So take all the first words. So here it's highlighted. So you can see that goodness gracious Father Franklin did go by pickling pumpkins and pies. So these first words you can take. So you can take G, F, D, G, B. So these are your first words and you can remember. So G means glucose. Then gracious is glucose 6-phosphate. Don't get confused. So whenever you're listening or you're listing down this G's, F and D's and all, get proper words or proper technical terms that is fitting into the option. So G is glucose, gracious. So gracious is glucose 6-phosphate. F is fructose 6-phosphate. So in this way you can remember. So glycolysis is getting ended over here. So this is all about glycolysis. So the next question is fluid mosaic model. So that's also a very important question that is surely going to come to your exams. So fluid mosaic model was proposed by the person that is S.J. Singer and Garth Lee Nicholson. So these are the two persons who invented or who discovered fluid mosaic model. And this model, what is it mainly explaining? So it's explaining the structure of the plasma membrane of the animal cells and the mosaic of such of more of the different components. So that is phospholipids, proteins, cholesterol and carbohydrates. And these components, what it is mainly doing? So these components is giving a fluid character to the membrane. So that is the main agenda or main idea that was done for this kinds of components. And each phospholipid, what are they having? They are having a hydrophilic head pointing outside. That is something related to water, and hydrophobic tail forming the inside of the bilayer. So this is a small thing about phospholipid layer and cholesterol. So what is this cholesterol? Cholesterols and proteins which are embedded. Embedded means which is inside the thing. In the bilayer is giving a membrane of a mosaic look. So here in this diagrammatic representation, you can understand. So a kind of mosaic appearance is given. So that is the mosaic look that is being, which is told here. And each component is having a specific function. So all these components of phospholipid, let it be proteins, cholesterol, carbohydrates, all they are having a kind of functions. They are having their own functions. So this is about fluid mosaic model. And next we are going to discuss about the main and very important topic, that is urea cycle. So what's this urea? So here you can see a kind of representation wherein you have to definitely or surely write your diagrammatic representation of urea cycle, and let's see what are the steps and what it is doing mainly. So what it is mainly doing, what is this urea cycle is doing inside the body? So it is removing excess nitrogen from the body. So when you're having a tabular column of like this, what's happening is that you are able to differentiate. So you can give your different headings as such in the colorful pencil. So how you can also, you can make your own notes so that in this colorful manner so that you can read. So you'll have an interest and you will have an idea, okay, what it is doing, this urea cycle, and you are able to remember the exact point. So in this way, when you're doing, you're definitely going to surely pass your exam and you will not have a fear, okay, I have not studied. So you'll have your notes with you so that you can take whenever you are free, you can read it. Okay, what it is doing. this urea cycle? Removes excess of nitrogen from the body. And where it is happening? It is primarily happening in the liver and to a lesser extent in the kidney. So mainly it is functioning in the liver and some parts of the kidney areas also. And how it is working? 6 enzyme and 2 mitochondrial transporters convert ammonia and aspirate and bicarbonate into urea. So that is how it is getting worked. So that is the main function that is happening in the urea cycle. And what it is mainly producing? So urea, which is relatively inert, soluble in water and easily exerted in the urine. So that is the production that is happening in urea cycle. And other names, there's even different names. So whenever you're getting a question in your question paper, you have to not get confused. So that is ornithine cycle, that's Krebs and Henseleit cycle. So these are the other names given to that, and the discovery was done by these two people. So that was the names given to the cycle. So it's also known as urea cycle, and different names are also given. So don't get confused in your question paper. So this is the same answer. You can write it for the urea cycle if they're asking these questions also. So don't get confused. So next question is glucose tolerance test. Okay, you think that this is not much important, but still, in case if they are asking, you can write something about glucose tolerance test. Okay, this glucose tolerance test is a test that is usually measuring the body's response to sugar. So that is also called glucose. So glucose test is also usually done, okay, to understand the sugar level in each person's body. And another name for this test is oral glucose tolerance test. In the last slide, we have discussed about the urea cycle. It is also having another name. Glucose tolerance is also having another name. So don't get confused. Write in different pages that about urea cycle one thing, you have to write about glucose tolerance test so that you're not getting confused about it. And this test can be used to screen. So which are all types? That is type 2 diabetes or pre-diabetes before any symptoms is occurring in the body. So either of the condition, you can do this glucose tolerance test. So in this way, this glucose tolerance test is helping, and it is used to find out whether the diabetes is causing existing symptoms or not. And more often, it is a type of version wherein the test is used to check for diabetes that is happening during pregnancy. And thus, that condition is called as gestational diabetes. So during pregnancy period, if the sugar level is high for the mother, then definitely that condition or that is referred to gestational diabetes. So that is about glucose tolerance test. So the next question we are discussing is about the steps of Krebs cycle. So what is this Krebs cycle and what it is mainly doing, and we'll see step by step so that you will understand it in a more clearer manner. And there is also a diagrammatic representation where you have to definitely write it in your exam, because it's a very important cycle. Even urea cycle is also important, and Krebs cycle. So in the Krebs cycle, acetyl-CoA, that's the oxaloacetate, that is converting into citrate. So what is happening? This acetyl-CoA from glycolysis, it is combining with oxaloacetate to form the citrate. So in this way, there's a conversion happening. So in each step, that's the process happening. And the key enzyme is citrate synthase. Next, you can see citrate is getting converted into isocitrate. Isocitrate, that is alpha, it is getting converted into alpha ketoglutarate. So again here, you can definitely read all these points, because what is happening, key enzyme, what it is producing. So in this manner, if you're going to a detailed study, then you're definitely understanding the steps of Krebs cycle. Here is the diagrammatic representation. This you have to read as well as you have to practice this diagram. It's a very important diagram. So that is also known as a TCA cycle. That is the cycle for Krebs cycle. If the question is even asked for TCA cycle also, you can write the same thing. Then in the next also, you can see alpha ketoglutarate is getting converted into succinyl-CoA, and succinyl-CoA is getting converted to succinate, and then succinase is getting converted into fumarate. So what is happening? What are the key enzyme? What it is getting produced? And fumarate into maltate, and maltate into oxaloacetate. So these are the steps that is getting happening, and next we'll throw this and we'll go to our next question, that is dietary fibers. So what is this dietary fibers? So you will get exhausted, so that is why we have done that kind of thing, so that next question will be a little more easier for you. So what is this dietary fiber? So this dietary fiber is a complex carbohydrate which is usually mainly found in the plants and in the human body that cannot fully get digested. And the dietary fibers is usually rough or bulk, so it is having many of the kinds of health benefits, including, so what are the help or what are the benefits that is usually done? That is digestion, weight control is happening in the body, blood sugar, heart health, and gut bacteria. So these are the kinds of things that is getting helped in our body itself. So digestion is happening in a proper manner, weight control makes you feel full faster, blood sugar that is getting slowed down in the blood, and there's a proper absorption happening which can help to prevent the blood sugar level increase. Heart health, that is boosting the heart's pumping capacity, heart health is in a very good condition. Gut bacteria feeds friendly gut bacteria. So this is about dietary fibers. And there are even few important points that you can also refer and write in your exam. So these are fetching you a good amount of marks. So next question is antioxidants. So that's again, you feel that okay, these are not important questions, but let's have a look about this antioxidants. So antioxidants, what are they? They are the man-made natural substances that is preventing or delaying some types of cell damages. So these are the things. So that is a small definition you can tell for antioxidants. And antioxidants are usually found having many kinds of foods, including fruits and vegetables, and they are also having available in dietary supplements. So there are some supplements that is helping in the body that are the antioxidants. Here you can see a diagrammatic pictures what in all are the berries, the leafy vegetables, all kinds of grapes, fruits, vegetables. These are kinds of supplement that is adding into the body, human body, and it's very helpful to the human beings. So vitamin A, vitamin C, vitamin E, all these kinds of vitamins are enriching in the body. So vegetables and fruits are most rich in a source of antioxidants. There's a good evidence having a proper, whoever is having a lot of fruits and vegetables, there's a research found that there's having a very kind of healthy lifestyle, as well as they're having low risk of certain disease condition is also found in the recent studies. But is it clear whether it is because of the antioxidant, something else in the food, or even other factors? So, high dose of these supplements of antioxidants may be usually initiated to the health risk in some cases. There's even some examples for that. High doses of beta-carotene, which is usually getting increased in lung cancer in smokers. So, there is a high chance of a risk in the lung cancers, especially who are smokers. So this kinds of things are also there. So there's also some different kinds of points also given. You can also refer that, and even you can refer your books so that you are able to identify and get more points and include in your exams. Then the last question here we are discussing is about the polysaccharides. So what's this polysaccharides? What is happening in the polysaccharides? There are having a lot of things in polysaccharides, but whichever is important to you is given here. The polysaccharides is the major classes of biomolecules. So this is your important key definition or key point that you can write in the starting. And they are having a long chains of carbohydrate molecules are there. So they are a there's very large extension of this carbohydrates composed of several smaller monosaccharides. So that's about the polysaccharides, and these are very complex biomacromolecules. They are usually having a lot of functions and very important source of energy in animal cells, and it is forming a lot of structural and even component of the plant cell. So these are the functions, or they'll have a basic and very large extensions you can see in animal cells as well as in the plant cell, and it is mainly having a homopolysaccharides or even heteropolysaccharides depending upon the type of the monosaccharides. According to the type of monosaccharides, they will be differentiated into homopolysaccharides and heteropolysaccharides. And these polysaccharides can be straight chain of monosaccharides, and it can also be the known as branched polysaccharides. So there are different types of polysaccharides, that is, polysaccharides can be categorized into two types, that is homopolysaccharides and heteropolysaccharides. Thus much, if you are writing, at least 2 to 2.5 mark is guaranteed for you. So this is all about polysaccharides. Hope you have understood the session. We'll come with more upcoming videos which is helping in your sessions, in your exam point of view will be coming. Stay tuned. Until then, thank you. Enjoy with EZI Nurse.